Good morning everyone and welcome to today's webinar. My name is Samuele Capomasi and I'm a process technologist at EMA Active Competence Center in Bologna. Today I'm glad to have with me Thomas Altman, Global Technical Consultant from Ecolab Pharma and Personal Care Division.
Thank you, Samuele and EMA, for having me here. And good morning to everyone attending. We really appreciate you take the time to join us. Today, Samuele and I want to show you a little bit of our daily work and share our practical insights into cleaning challenges in the pharmaceutical industry.
Cleaning isn't just a routine task. It's a critical part of ensuring product quality and compliance with stringent industry standards. Together, we will dive into some of the toughest cleaning challenges we face in the pharmaceutical industries, and we will share practical approaches and solutions drawn from our daily experience.
As a global technical consultant, I'm often asked what kind of cleaning processes, cleaning procedures should be used. And my answer is generally always the same. It's depending on the residue. So it's really to know what kind of residue we need to remove and what kind of critical cleaning process parameters you have. For all cleaning activities, we have basically 4 cleaning parameters, and they are described in the so-called Zinner circle. Each effective cleaning requires some time, a certain kind of chemistry. Also, we need sometimes only water as chemistry, and we need the temperature and the mechanical action. Even the GMP guideline, Annex 15, is outlining to us we should know what kind of process factors are variable and what are affecting the cleaning effectiveness and performance. To determine the critical cleaning process parameters, Ecolab has different cleaning process development studies established. The results of these cleaning process development studies show cleaning chemistry, what is working best, and also the temperature, what needs to be the cleaning at. At least also how long should a cleaning process take will be visible. To determine the critical cleaning process parameters, Ecolab has the process development studies over the past years, starting with a simple test on solubility. And if we need a little bit more mechanical action, even beaker studies are performed.
Also at our EMA Active Competence Center, with the support of Ecolab, we perform coupon studies to evaluate the compatibility of the material employed in the equipment in order to give technical support to our internal validation team. This enables to guide proper maintenance and ensure material integrity over time.
The result of cleaning process development studies Ecolab performs show that cleaning chemistry is working best at what temperature. How long should the cleaning process be will also be outlined. Also, a consideration on all of the 4 factors for cleaning is outlined in our study report. In the example shown here, you can easily identify that the temperature is driving the cleaning. The concentration of the cleaning agent is maybe not that driving the cleaning, only the temperature. For other products, the cleaning parameters could be completely different. This is why I said at the beginning directly, it's always depending on the residue what kind of cleaning we need to perform. In the pharmaceutical manufacturing, we have cleaning validation to take into account, and as we have now some insights for you how to perform cleaning processes with hard to clean residues, I would like to hand over to Samuele to show how effective cleaning processes are installed in the EMA equipments.
In this first practical session, we will be sharing with you some of the real-world cases from our competence center, showcasing hard to clean materials and the solution that we have found to remove them. We will explore some of the most challenging cleaning scenarios in the pharmaceutical industry, discussing the solution that we have used to address them. Let's start with cellulose, a widely used material in pharmaceutical industries, both in granulation and coating process. The cleaning approach depends on the type of cellulose, but in general, we have found that hot water combined with an acidic detergent, such as CIP72, is the most effective. Strictly connected to this topic, another challenging material to be cleaned is the derivatives of the methacrylic acid. These type of coatings, material like eudragit, are designed to protect the active ingredients from stomach acid or to control the release over time. Due to their robust and durable nature, these coatings can be challenging, but good results can be achieved thanks to proper knowledge of the material. In general, it is important to avoid high temperature, as the coating can become soft and pliable if exposed to excessive heat. These examples show a validated cleaning cycle for a coating pan, in which eudragit S100 was present. For this type of eudragit, a solution prepared with a basic detergent, such as CIP96 at 2%, works effectively to break down and remove the residues.
Thank you, Samuel, so much for this practical insights. So we just learned that the right cleaning process can give you a good cleaning result even for challenging materials. So cellulose, for example, was touched, as well as the complex topic eudragit. So eudragit is really a big variety of different methacrylic acid derivatives, and they are not so easy to clean if you don't know what certain pH range they are solubilized in. And we have even a more harder to clean solution if we talk about sustained release coatings. So they are even pH independent, not removed. They need to be solubilized with a product like Cosapure 81. Generally, we see often cleaning application for eudragit coatings happening at hot temperatures, and Samuel just outlined. It should be at lower temperatures, because then we have a better cleaning effect, and often also the additives like pigments do not stick so hard on surfaces if we clean at 30 degrees. An optimization on sustainability is also going into this direction. So we see today a lot of cleaning processes have been performed over years at hot temperatures, but we reduce it now to 30 degrees also to save energy. These low cleaning temperatures are not always the solution. And Samuele will give us now an example on, for example, iron oxides, where we need a hot temperature for cleaning. And you may guess how to identify what is the best temperature. Of course, the cleaning process development studies. So let's have a look what Samuele outlines to us now, how EMA is dealing with the topic on iron oxides.
Now, let's talk about iron oxides, commonly used as colorants in tablets and capsules because of their stability and intense pigmentation. The challenge with this material lies in their strong adhesion to stainless steel surface. They form highly stable layers that are tough to remove with standard cleaning methods. We encountered this challenge in a pilot-scale high-share mixer granulator, where iron oxide was one of the major components of the formulation. After production, reddish stains were clearly visible in the internal surface of the granulator, such as gaskets, joints, and other less accessible areas. Optimization of cleaning process was needed to improve the removal of any residues. In addition, we used an acidic detergent such as CIP77, designed specifically for the removal of iron oxide pigments, which contributed to enhance the overall cleaning efficiency. This case highlighted how standard cleaning strategies often need to be adapted when dealing with specific materials like ferric oxide.
I would like to show you now what can happen if a non-successful cleaning is happening. So you can see on the picture that there is a film on the stainless steel of the pigment. This situation is what you maybe already know if you clean and deal with iron oxide pigments. You can get a complete colored surface and the pigment is all over the place in a fine film. Often a redeposition also happens to the equipment, and this is especially true if you have long cleaning cycles with alkaline materials. So these long cleaning cycles should be avoided to not have the separation of the pigment out of the matrix of the coating or product. So we often recommend to go for short cycle times and short cleaning cycles and not use long cleaning applications. Nevertheless, the most effective way for us is first of all to remove the residue, for example the coating material, and then an acidic step like we just heard, COSA-SIP 77, to remove the iron oxide residues should be performed. Looking now to another example and a pain point really coming from the pigment area, this is titanium dioxide. And for those of you who have iron oxides already now ticked the box, there is another challenge with titanium dioxide, and let's see what Ima is doing and Samuele will explain to us.
Titanium dioxide is widely used in coating processes due to its unique functional properties as whitening agent and opacifier. These characteristics not only improve the appearance of the final product, but also protect light-sensitive active ingredients. The difficulty with titanium dioxide lies in its very low solubility in water and its strong tendency to adhere to metal surfaces. Over time, it can form chalky films resistant to removal, especially when drying occurs before cleaning begins. In our experience, the most effective approach involves the use of an acidic detergent, which helps in breaking down and loosen the residues, facilitating its removal from equipment surface. A key element in the cleaning strategy is timing. Starting the cleaning cycle shortly after production helps prevent drying and adhesion of the coating residues. Also, using design spray system ensure that all areas prone to accumulation are adequately treated. In this case, a high pressure system that can reach up to 5 bars helps detach particles from surfaces, hitting the target due to its mechanical action. Successfully removing it requires a tailored approach that combines appropriate chemical action with control process parameters to prevent the buildup and ensure compliance with cleaning validation standards.
After this practical approach now we have seen from Samuele, we want to look also how do we see in our cleaning process development studies some residues and maybe even hints on the cleaning we get from the laboratory experience. So the formulations to be cleaned can really break apart, and that is what we have also seen with the iron oxide cleaning. So also here it applies. Long cleaning cycles can break apart the formulation and titanium dioxide can stick on surfaces. This is what we just heard. Therefore, for us the most important is the temperature, not more hotter than 65 degrees C, and short cleaning cycles. So that's fairly comparable to the iron oxide residue, also to get rid of titanium dioxide. Let's finally have now a look to another topic not related to pigments. It's more like a processing aid, and we want to have as last example to talk about stearates. And I would like to hand back to Samuele to get to know what Ima is doing for stearates cleaning.
Stearates are widely used in a tablet and capsule formulation. Due to its waxy and hydrophobic nature, it tends to adhere to metal surface, and its removal it can be difficult with just water or general purpose detergent. We tackle this issue by using high water washing cycles combined with an alkaline detergent. Using a product like CIP92 or a similar high-performance alkaline cleaner can be suitable as it can emulsify and break down this kind of residues. In this case, a pre-rinse with hot water is useful to soften and loosen the residues. Then we apply an alkaline cleaning solution, allowing sufficient contact time. Even in this case, mechanical action given by water pressure is the key for the success. In the end, a final rinse with demineralized water is needed to remove all detergent residues.
This was a very clear way forward how to deal with stearate residues in the equipment. What I would like to show you now is also a little bit on how these process residues look like in the laboratory equipments and tests. So you can often see hydrophobe surfaces if you do not see the white powder of the stearate. So these hydrophobe surfaces are visible with droplets, and this is really something where we would say a not adequate detergent was used. The most effective cleaning was described now by Samuele, and I just want to remind you that we need hot temperatures for the cleaning as well as the appropriate cleaning agent containing to remove and to get this surface really clean.
The example we share today represents real challenges that we have faced and the solution that we developed to overcome them. What we presented isn't just a list of cleaning methods, but a collection of experience that can serve as a guide when you tackle similar issues in your own work, from laboratory scale to large-scale production equipment. Understanding the properties of each material and customizing the cleaning process is the key to success.
So we hope that with all our examples, we did come to the conclusion for you as well that a cleaning in pharmaceutical industry is not just a straightforward solution. We really need to look what kind of residues are present, what kind of products are used, and also what kind of process parameters can we tackle. And now I would like to hand over the last time to Samuele and thank you for your attention. I hope you got a lot of good information out of this event.
Thank you for your attention also from my side, and thank you, Thomas, for joining us today. I'm looking forward to hearing about your own experience in the field and hopefully see you soon.